Hi everyone, and welcome to our video on genetic engineering. This is part of our Module 6 Part 3 video series looking at manipulating genomes, and in this video we'll be having a look at the specification references 6.1.3 F Part 1, the principles of genetic engineering, and 6.1.3 F Part 2, the techniques used in genetic engineering. First thing then, what is it? What do we mean by this phrase genetic engineering? And before we delve into its actual definition, we should make ourselves aware that it goes by multiple names, just because biology's fun like that. So whenever you see genetic engineering, recombinant DNA technology, or genetic modification, it's all the same thing. And basically what we're talking about here is the manipulation of DNA sequences in an organism through some means. So how can we actually do this? What makes it possible that we can take the gene from one organism and stick it into a completely different one? And the simple answer is DNA is universal. And what we mean by DNA being universal is that all life forms here on Earth have DNA. And that DNA is going to be made of the same four bases: adenine, thymine, cytosine, and guanine. But in addition to that, the way that those bases are arranged into those triplet codons that code for an amino acid, that is universal too. So when we've got a codon, it's going to be coding for the identical amino acid in a human as it does in an insect. If we now move on to the actual process of genetic engineering, there are four key stages to this. The first one, we need to obtain the required gene. Secondly, we insert a copy of that gene into our vector. The vector is then going to be inserting that gene into our host cell, and then finally the recipient will express that novel gene. Let's go deeper into each of those four phases then. So first up, obtaining the gene. And we've got a variety of ways that we can actually get our hands on the gene we want. So the first way is that if we obviously know which cells are expressing a particular gene, then we know that those cells are going to have copies of the mRNA for that particular gene present. So what we can do is we can isolate the mRNA from those cells, and then we can synthesize a strand of cDNA using the enzyme reverse transcriptase. Now, reverse transcriptase is an enzyme that is going to be using the mRNA as the template and make a copy DNA. So we've got our cDNA being made from it. Obviously the normal process is that we use DNA as the template and make the RNA, but we're doing this in reverse, hence reverse transcriptase. Once we've got our cDNA, what we'll use are some primers, some DNA polymerase, and then we will actually add those to the cDNA to make that double-stranded length of our required gene. Option 2 is we can use a DNA probe, and that's going to locate our specific gene. Once we identify where it is, we can cut it out using restriction enzymes. Third option is if we know the nucleotide sequence, which we will have obviously worked out through DNA sequencing, then we can use an automated polynucleotide synthesizer to synthesize the DNA. Now that's basically a fancy way of saying a machine that makes a bit of DNA artificially in the lab. So automated, because obviously it's computerized, polynucleotide, many nucleotides, synthesizer, making. So all we're doing is we're making a bit of DNA in the lab using a sequence that we've discovered through that DNA sequencing. Or finally, we can obviously create our PCL primers designed to amplify the gene from any genomic DNA if, again, we know that sequence. The second stage is using this vector. So once we've got that gene that we have for our desired characteristic, whatever it may be, we've got to have some kind of technique to get it into the host cell. And this is where a vector comes into play. The most common vector that we use in genetic engineering are plasmids. So our plasmids are those little circular loops of DNA we find in our prokaryotic cells. And if we take a plasmid, combine it with restriction enzymes, they will cut it at those recognition sites. Now, they're going to create something called a sticky end, which is basically where we've got some exposed bases. We then use our complementary nucleotide bases to add to the end of that gene so that we can then anneal it onto the plasmid when we mix it with DNA ligase. And hopefully we remember DNA ligase is the one that does the joining. So we've encountered that before when we're looking at DNA synthesis, where we talked about the Okazaki fragments from obviously that moving in the reverse order 3' to 5' end. Then we're making these short little fragments that have to be joined together. DNA ligase is the bit that joins it. Same idea here. We've got a sticky end on our plasmid. We're then creating the complementary sequence on our inserted gene. And then, in the presence of DNA ligase, they're going to join those two things together. An alternative method to the plasmid is we can use an attenuated virus. And the way that that works is a virus is going to insert its genetic material into the host cell. So it has that mechanism for actually inserting genetic material into a host cell anyway. If we attenuate the virus, we're obviously weakening it, so it's not going to cause that infection, but it will insert our intended gene into the host cell. Once we've actually got our gene into our vector, we've got to get that vector into the host cell. And again, there's not just one way to do this. Obviously, the way they mention on your specification, first of all, is this process of electroporation. Now, as soon as we see electro, hopefully our brains are thinking electricity, and poration is basically creating little holes. So what we do in the process of electroporation is we apply this high voltage pulse to the cell, and when that happens, it disrupts the membrane, which means the plasmid can basically sneak through those little gaps that have formed. So what we've got, electroporation, high voltage pulse, disrupting membrane, and then the plasmid enters through a process called transfection. We do have alternatives though, and first one is this heat shock treatment. So if we're using something like bacteria to produce human insulin as a good example here, we've got our plasmid that we want to be taken up by that bacteria, and we'd expose them to calcium chloride. Now, what we're going to be doing is we're going to cycle that between 0 degrees and 42 degrees in the presence of that calcium chloride, and that process makes the bacterial walls and membranes more porous. So what we're seeing here is the calcium ions surround that negative DNA and the phospholipids, and as a result of that, we reduce those repulsive forces, because obviously both of those have got negative charges, so ordinarily they'd be repelling each other. But if we've obviously surrounded them with our positive calcium ions, then they're no longer going to be repelling in that way. So they're more able to enter the bacterial cell. We could also use these things called T1 plasmids. And this is where we're going to use these in our Agrobacterium tumefaciens. And basically, this is then able to infect plants. So what we're doing here is we use our T1 plasmid, we insert our desired gene into that. That is then inserted into Agrobacterium tumefaciens. And then we expose our plant that we want to take this up to the Agrobacterium tumefaciens, and it will insert that genome into the host cell genome. There is one other method that we can use, which is a direct method. And we'd utilise this for any of those plants that aren't susceptible to Agrobacterium tumefaciens. And it's a device called a gene gun. Now, it sounds quite ridiculous that we have a gun that fires genes into things, but yes, we do. And we can see it at the picture at the bottom there. This thing here is a gene gun. And basically what we're doing is using these small pieces of gold or tungsten coated with our desired DNA, and we literally shoot it into the plant cells using our lovely little gene gun. So you can see we place it over the leaf, and then literally we're going to shoot it in on teeny tiny pieces of precious metals. The next thing we really need to make ourselves very aware of are the enzymes involved in this genetic engineering process and what they do. So there are 3 key enzymes that we need to know about: reverse transcriptase, restriction enzymes, and ligase enzymes. Reverse transcriptase, first of all, which is obviously one that we've mentioned in this video, but you in theory have heard about back in module 4 where we looked at HIV. Now, reverse transcriptase is going to be used to produce that cDNA from the RNA templates. And this is that process of reverse transcription, starting with an RNA template and then creating cDNA. Second type of enzymes, the restriction enzymes. These are obviously types of enzymes that occur naturally in bacteria and archaea, and they use them as a defense against the phage viruses. So basically they're using these enzymes to protect themselves from infections from a virus. The reason that we can use these enzymes in bacteria and archaea, but they're not affected by them, is that prokaryotic DNA is protected due to methylation at those recognition sites. So what we've got is this methyl group blocking the recognition site, and therefore these enzymes can't actually carry out their action. The first one of these restriction endonucleases that we obtained was from E. coli, and we very creatively called it EcoR1. So you can see, here's its little name, Eco for E. coli, R1, restriction endonuclease number 1. When we're talking about these restriction enzymes, we basically have two options. Option 1 is a restriction enzyme that makes a sticky end through the staggered cut. So where we've got our double-stranded DNA, it's going to actually cut it so that we have a longer piece on one of the strands than the other. So that means we've obviously got exposed bases ready to obviously join to another piece. Other ones just make a blunt end, so when they cut it, it's literally the same length at that point. But the key thing about either of these types of restriction enzymes is they're recognizing a palindromic sequence. And when we talk about palindromes, these are sequences that read the same forwards as they do backwards. Our third and final enzyme that we need to know about is the ligase enzymes. And if we're ligasing or ligate something, then this is going to be joining it, okay? So what we are seeing here is the ligase enzymes are going to be joining these DNA fragments through catalyzing condensation reactions between the sugar and phosphate groups. And hopefully we remember sugar and phosphate groups are going to form those phosphodiester bonds to form that sugar phosphate backbone. Let's have a look at a specific example then, and we're going to go with an old favourite that you've probably seen at GCSE as well as A-level, human insulin. Now, the first stage, we need to get hold of our gene, and hopefully from our module 5 knowledge, we remember that insulin is made in the pancreas through the islets of Langerhans. So what we're going to do is we are going to extract mRNA for insulin from the islets of Langerhans in the pancreas. Once we've got that mRNA, we are then going to mix it with the reverse transcriptase to make our cDNA. As soon as we've got that cDNA, we add our DNA polymerase so that we can create this double-stranded part of DNA. So basically we've got a double-stranded DNA copy of our desired insulin gene. What we then do is we're going to attach these unpaired nucleotides onto the end to make those sticky ends which are going to be necessary when we join it to our vector. We then take the plasmid from E. coli and we're going to mix it with the cDNA and the DNA ligase to form those recombinant plasmids, after obviously we've cut that plasmid using the restriction enzymes. Once we've got a recombinant plasmid, what we're able to do is mix the new E. coli cells with the recombinant plasmids, and we give them a heat shock treatment so that they take those plasmids up. In theory, we've then got our genetically modified bacteria who are going to then happily produce human insulin, which we can then extract. Now, obviously it'd be lovely to just believe that this process is completely foolproof and 100% guaranteed to work every single time, but biology is not like that. You know, if you've done experiments with any plants, they don't always do what they're meant to. So we'd be pretty foolish just to trust that our genetic engineering had been perfect. So how do we check it then? How do we make sure that our desired gene is where it should be? And the answer to that is because we're utilising this plasmid as our vector, we can actually use some of the other features of that plasmid as a checking mechanism. Now, bacterial plasmids typically have antibiotic resistance genes present within them. So what we're going to do to make sure that that cDNA of our desired gene has been incorporated into the plasmid is we're going to insert it into the middle of a resistance gene for a particular antibiotic, such as tetracycline. That plasmid, though, is going to contain other resistance genes. And so we may very well have a second antibiotic resistance gene for something like ampicillin present. Because we're not inserting the cDNA into that particular gene, it will retain that resistance. So once we've gone through this process of, in theory, inserting our vector into our bacterial cells, we then just grow the bacteria on agar plates with ampicillin present. Now, the first thing that we're going to see here is that any bacteria that do not have the plasmid will not grow, because any that don't have that plasmid do not have the resistance gene for ampicillin, so they'll be killed by it. Any of the bacteria that do grow will have taken up the plasmid, but that does not mean it is a guaranteed recombinant version, because remember, we didn't disrupt that in any way, shape or form. It just says the plasmid made it in, not that our intended DNA sequence is part of that plasmid. So we need a second stage here. Once we've done that first plating, we're then going to carry out what we call replica plating onto a second agar plate with the tetracycline antibiotic. So what we're going to do is we're going to select those colonies that are growing on our first agar plate, and then we're going to grow them on a second agar plate with the second antibiotic. Now, this time, what we're going to find is that it's only going to be those without the recombinant plasmid that are growing. Any of them that took up our desired gene will have that inserted into the middle of that tetracycline gene. So that means they're not going to be resistant to it anymore. So once we know which ones have got the recombinant plasmid, we can then extract those particular colonies from our first plate and then grow them in large numbers. We can check this process using two other methods, which are fluorescent markers. So we're again going to be attaching a fluorescent marker to our particular sequence, and they will just glow under ultraviolet light, so you can select the right colonies based on that. Or we could use enzyme markers. The final thing we're going to consider in this particular video is a bit of safety. So when we're thinking about creating genetically modified things like bacteria, etc., we've got to be sure that what we're doing in that lab is not going to create issues in the wider world. So if we're creating transgenic bacteria with antibiotic resistance, we don't want other bacteria in the wider world getting antibiotic resistance genes. That's bad. That means less of our antibiotics are going to work against common illnesses, for example. So we've got to stop them from being able to escape the lab. And that doesn't mean that we've got to have some extreme lockdown procedures, etc., like you see in these weird films. Obviously, there will be procedures, but we design the bacteria a specific way. And what we do is we have what we call a specific gene knockout. Now, what we're doing here in the design is we're actually going to make sure that the bacteria we are using, these transgenic bacteria, cannot survive outside the lab. Now, if you've seen Jurassic Park, the first one, many, many years ago, then they had this same idea that they used in that film. So their whole idea was that these dinosaurs couldn't actually escape from the island, etc., because they needed a particular nutrient that the actual researchers supplied to them, because they'd removed the ability to synthesize it from their genome. Same kind of idea here. What we're doing is we're removing that gene that's going to allow the bacteria to synthesize a particular nutrient. So unless they're being provided it by people in a lab, they won't be able to make it, they will not survive. This is working better in our transgenic bacteria than it did in Jurassic Park, which is good to know. As always, I hope you found this video useful, and of course don't forget to subscribe to the channel so you can see when the next video is uploaded, and then head on over to the website where you can find a range of other resources that will help you in your A-level biology studies.